Hello, students. Today we have to learn about the monoclonal antibodies. What are monoclonal antibodies? Monoclonal antibodies are those antibodies which are produced by same kind of cells, and they these antibodies will be a homogeneous mixture of same type of antibodies which will bind with the same epitope. It's a very good tool. So we start with this monoclonal antibody production, and my name is Dr. Zahir Hussain. So in monoclonal antibody production, we see the first thing is that we should know that for which protein or epitope we have to produce monoclonal antibodies. So first we should have this purified protein, and then we will add adjuvant with it, and then we will immunize the mouse. And then we will get primary response, and then after primary response, we will get the secondary response. And after getting secondary response, we will isolate the spleen, slaughter the animal, and then take out the spleen from that immunized mouse. And the spleen cells, because the spleen is a secondary lymphoid organ and it have more number of B lymphocytes. And B lymphocytes are the lymphocytes which are producing antibodies. So we have to sacrifice the animal, and we'll take out the spleen and we'll macerate it and then isolate B lymphocytes. Then we go for the fusion of these myeloma cells, and these myeloma cells are HGPRT minus. So myeloma cell, what are myeloma cells? Myeloma cells are cancer cell line of these B cells, and it's a special cell line because the gene for HGPRT is knocked out. It's not working. And this HGPRT gene is required for the synthesis of nucleotides. So we will fuse these B cells, which we have isolated from the spleen of that immunized mouse, and we'll fuse it with the myeloma cells with the help of this some liquid, or maybe with the help of electric current. So after fusion of these cells, that is B cells and myeloma cells, which are HGPRT minus, then we go for formation of hybridoma cells. The fusion will take place, of course, it will be the hybridoma formation. The more important thing than this is selection process. Selection in het media, only few cells will survive. What is het media? Het media, you can see it is made up of three alphabets. H stands for hypoxanthine, A for aminopterin, and T for thymidine. So het is composed of hypoxanthine, aminopterin, and thymidine. So this media is termed as the selection media. So how this will work? Selection process. So we know that there are two pathways for the synthesis of nucleotides. So there are two pathways. One is de novo pathway and other one is salvage pathway. So here we see that sugars and amino acids, they will combine and then through this pathway, they will make nucleotides. Another one is salvage pathway in which this hypoxanthine and thymidine, they will combine and they will make these nucleotides and then DNA. So here we see that in this de novo pathway, there is an enzyme which is involved, that is DHFR, dihydrofolate reductase. And aminopirin is the inhibitor of dihydrofolate reductase. If we add this in the media, aminopirin, then the synthesis of nucleotides will not be possible through this de novo pathway. So in salvage pathway, we know that we are using this myeloma cells which are HGPRT minus. It means myeloma cells cannot synthesize this nucleotides in DNA with help of this pathway, salvage pathway, because they are not having this HGPRT enzyme. While in de novo pathway, we see that on the het media, these myeloma cells and myeloma cell fusion products or hybridomas will not be able to produce colonies. So basically, purpose of hybridomas selection is that we want to select the hybridomas, but which one? Because when we will add or will fuse these cells, so we will get three types of fusion products because we have two types of populations in it, B cells and myeloma cells. So either myeloma cell will fuse with myeloma cell or it will fuse with the B cells or B cell will fuse with the B cell. So since it is difficult to culture B cell fusion products, so it will not be a problem. But the problem will be that myeloma cells fusion products can grow there. But if we grow it on the het media, this HGPRT minus myeloma cells cannot utilize salvage pathway for the nucleotide synthesis and DNA synthesis. So they cannot grow there on the het media because they cannot utilize salvage pathway due to the fact that they are HGPRT minus, and they cannot utilize de novo pathway because aminopterin is inhibiting this dihydrofolate reductase so that nucleotide synthesis will not take place. And then if there will no DNA, there will be no cells and there will be no colonies. So we see that only B cell myeloma cell fusion products will be able to grow on the het media because they have HGPRT, because B cells were having HGPRT, they will utilize this pathway. They cannot utilize this de novo pathway, but they can utilize salvage pathway, or therefore on the petri dish, we will get colonies, and these colonies will be of hybridomas, which are hybridomas are the fusion products of these myeloma cells with B cells. So screening for desired antibody producing hybridomas. So what is our desired antibody? So we already have defined that we should have that purified protein against which we want to produce monoclonal antibodies. So screening, we will screen for that desired antibodies, and those hybridomas which will have that desired antibody, then they will be selected and they will be taken for further study. So cloning of positive hybridomas, for example, limiting dilution. So we will clone, we will grow, and when we grow these hybridomas, it means we are cloning the hybridomas. So cloning of positive hybridomas, that those hybridomas which are positive, with the help of this limiting dilution technique, we can clone them and isolate them and purify them. So then expansion of cloned hybridomas will take place. We will grow them in more amount in large fermenters. The production of monoclonal antibodies, it can be in vitro or in vivo. So it can be in vitro or in vivo. In vitro means bioreactors and culture flaws, and in vivo means in the peritoneal cavity of the mouse. So it's better to go for the bioreactors because animals will not be involved and there will be no worries about this zoonotic diseases. And also animals will be not involved because we need thousands of animals and it's not ethically right to kill so many animals and utilize them for your experimentation, even if we have some other route, that is bioreactors. And of course, in bioreactors, we can produce more antibodies because in the mouse, there can be a small space, a peritoneal cavity, and in the bioreactor, it can be thousands of letters. Then we will purify monoclonal antibodies with the help of affinity chromatography. So in the chromatography column, we can use that protein of interest against which we have produced monoclonal antibodies as a ligand or coating material through, and then the antibodies will bind with that, and then we can elute it and we can get it in the purified form. And then we will characterize it, and after characterization, we will store it for further use. And we know that most of the monoclonal antibodies are used for the diagnostics of different diseases, for detection of bacteria, for detection of viruses, for diagnosis of cancer, and to follow the treatment of the cancer, and also to treat the cancers. So there are many more uses of these antibodies, and it's a billion-dollar business. So, okay, thank you.